So this is an experiment to show how did they figure out it was a triplet code. Okay, so RNAs are read in sets of 3. So the AUG is the first codon that codes for methionine. And then the ribosome reads in sets of 3. The next 3 code for an amino acid, the next 3, the next 3, the next 3. I'm not going to go through the experiment as to how to figure out, okay, it's not a 1-to-1. Well, that one's easy. Okay, if you have, we have 4 base pairs, right, in DNA, and we have 4 nucleotides in RNA, right? So you can do a direct, right, A's and U's, G's and C's, C's and G's, T's and A's, right? That's a 1-to-1. How many amino acids do we have out there? 20. We have 4 nucleotides in our mRNA. Can it be 1-to-1? Can it be an A goes with 1 amino acid always? No. There are 20 of them. Otherwise, we'd only use 4 amino acids. We'd have 4 amino acids. We don't. We have 20. So there's no way we could just, it could be a 1-to-1 like it is for replication and transcription. Right? It has to be something else. So I did a whole bunch of experiments to figure out, is it 2? No, actually, that's not even enough to get up to 20, unless we didn't use all 20 amino acids, but we actually do. And then the 3, yeah, you have way enough, 64 possible triplets. That's definitely enough to cover 20. Okay? But it's still not going to be then, 1 codon to 1 amino acid, because we have 64 possibilities and only 20 amino acids. So some of those stupid codons are going to have to code for the same amino acid as somebody else. Right? Does that make sense?
But the first 2 always stay the same.
Almost always, yeah. So we'll say that. In the codon, right? So we have to have maybe 3 codons equals 1 amino acid, because there's 20 amino acids and more than 60 codons. Okay, so it can't just be, except for some of them. So AUG always is methionine, and that's the only one that codes for methionine. But there are other codons that, there's 5 different ones. 5! I don't even know what I'm talking about. Different codons that code for the same amino acid. Okay? So that's all this says. The triplet code is redundant. It's not 1 code, 1 amino acid. And that's the rule. Because otherwise we'd only have 20 different triplets. We actually do use all 64 triplets. Okay? But each codon does just specify 1 amino acid. Right? So if it's UUU, it always codes for the exact same amino acid. Somebody else might also code for that amino acid, but any individual codon can only call 1. Okay, it's specific for that. I'll show you the little thing. And then, of course, the start is AUG, and we have 3 different stop codons. So there are 3 triplets that code for no tRNA. They stop. That stupid protein comes in and blows the shit out of the ribosome. Okay? So really, we have 61 codons that meet up with the tRNA. Because 3 of those codons are stops that are only that release protein. Okay? So how the hell does that work? Here's the stupid genetic code chart. It was figured out, again, experimentally, just like everything else is. And the book walks you through those experiments so you can nerd it up and read through that and see how they did it, and it's actually pretty good, right? Somebody was pretty smart and figured that shit out. But we're not going to walk through it, okay? But this illustrates our deal, right? If we look at leucine, there's 4 different codons that all code for leucine, okay? And then if you look, let's see, they might show up. Let me find one that is in multiple places, right? Is leucine another? Oh, right up there too. Yeah. So there's 6 total. Okay? If you look at any one of those individual codons, so CUU, it does not exist any place else on this chart. It only, always codes for leucine. So each codon is specific for 1 amino acid. Each amino acid can be coded for by more than 1 codon. Right? You could see it. Leucine has 6. It's not one codon to one amino acid. That amino acid can, got 6 options. But poor each codon only stuck with his one amino acid. Okay? And if you look, which I don't even know where it is on this chart. Where's my friend? Where's my friend? Here's methionine, right? It's AUG. It's the only thing, that is a direct one-to-one. One codon codes for methionine. Methionine can only see one codon. But most of them are slutty, right? Most of the amino acids are pretty sleazy. Right? Tryptophan's prude. I don't know. And then you can see the stop codons up there in the corner. Those guys, only that release factor sits there, no tRNA, no amino acid. Okay? For the exam, you will get this chart to answer any questions I ask you about codons. Right? I can tell you, you have, this is your codon, what amino acid does this code for? And then you look at the chart and you go, alanine. Okay. I don't think there's any class here at CSUN that you have to memorize this chart. Okay, you'll get the chart in 380, you'll get it in 360. You need to be able to use the chart and understand what all that means, but you'll never be asked to memorize it. If you end up doing research on lots of proteins and you're doing mutations and amino acids and switching codons, you end up by accident memorizing some of them. And then it's easier than going and look at that stupid chart every time you need to do something. But there's no time in your life in science where you have to memorize stuff like this in the real world. So we try not to make you memorize it just for something to memorize. Okay, but you have to be able to use it. And hopefully we'll have time to talk a little bit about that, about using it. Okay. Then we need